Okay, doctor.
Okay. Assalamu alaikum wa rahmatullahi wa barakatuh. I am Dr. Arij al-Mugheri, consultant hematopathologist and head of hematopathology division in King Abdulaziz Medical City, Riyadh. Good evening and welcome everybody to SSBMT webinar series. I would like to thank Dr. Al-Askar and SSBMT team for the invite and organization. Also, I would like to thank the sponsor for tonight's talk, Astellas and Janssen Company. It gives me a great pleasure to introduce our speaker tonight, Dr. Saeed Al-Turki. Dr. Saeed is a consultant molecular genetics, genomics, and bioinformatics. He is the head of translational pathology lab and molecular pathology in King Abdulaziz Medical City, Riyadh. Dr. Saeed obtained his PhD degree in genomics and bioinformatics from University of Cambridge. Also, he completed his clinical fellowship in molecular genetics and genomics in Harvard University. We are looking tonight, to hear from him about diagnostic approach to myeloid neoplasm using NGS. With that, I will turn it over to Dr. Saeed. Dr. Saeed.
Assalamu alaikum wa rahmatullah. Good evening, ladies and gentlemen. Thank you very much, Dr. Arij, for the kind introduction, as well to the SSBMT kind invitation, Dr. Al-Askar, my esteemed colleagues. It's my pleasure and honor to be here today to speak about the NGS role in myeloid neoplasia. So please let me share my screen. Dr. Arij, can you confirm you see my screen?
No, not yet. No, we can't see it, Doctor. Can you? Okay.
You see it now?
Yes,
okay.
now.
Excellent. Thank you so much. So this is my disclosure. And today we will be speaking about the NGS when it comes to the bone marrow initial workup in myeloid neoplasia. And the recommendation from WHO, European Leukemia Network, NCCN, and ESMO is to go through morphological assessment, as you all know, combined with flow cytometric immunophenotyping, and also cytogenetics, karyotyping, and FISH, as well as molecular studies in order to provide the required answer for diagnosis, prognosis, as well as delineating the risk for the patients. And one of the goals of this talk today is this really great paper that was published a couple of weeks ago in NEJM, which is titled as Genome Sequencing as an Alternative to Cytogenetics Analysis in Myeloid Cancers from Washington University by the group led by David Spencer. And this paper, I think, has a lot of, made a lot of noise simply because it is promising to merge or at least do the integration between cytogenetics, karyotyping, FISH, and the molecular study in one test in an extremely fast fashion within 5 days with a reasonable cost. So the goal today is to see how realistic is this paper when it comes to the clinical practice outside large institutes with a huge infrastructure for genomics. And together we will go through some of this paper as well as our experience at National Guard hospitals with some more realistic, let's say, adjusting the expectation through this, through the local expertise in the region. So testing for myeloid neoplasia is considered complex today, as you can see by this string. And this paper is trying to help us take this complexity of testing from the left to the right side. So the first question comes to mind is why it is complex. Where does this complexity arise from? And we have a few factors to discuss in order to understand this complexity. The first, as you all know, is the heterogenic genomic alteration landscape for myeloid neoplasia. So we are looking at very different classes of biomarkers. Here we are talking about single nucleic variants, for example, the JAK2 mutation, the insertion and deletions. One of the examples is FLT3 ITD mutations, copy number variants, the deletion and the duplication of very large segments of the DNA, structural variation or translocation between different parts of the genome. And here is an example of BMR-RARA fusion, as well as clonality or the clonal assays for the immunoglobulins. So all these biomarkers play some role in different myeloid neoplasia. And in the lab behind the scene, we have many methods to use to detect these biomarkers. One of the most commonly used and old techniques is the reverse transcriptase or the real-time PCR, which is able to detect different classes of biomarkers. FISH as well is really good for copy number variants and structure variation. MLPA is another method that's mostly for copy number variation and slightly for the point mutation. Capillary sequencing, arrays, as you can see, is only for copy number variants mostly. And the NGS, next generation sequencing, which I put in red because it has the promise to actually be so good in detecting all these biomarkers, at least theoretically. And then to add to this complexity, we have a plethora of biomarkers, and some of them will be used for diagnosis, some will be used for prognosis, others will be used for targeted therapy selection. So now we have a many-to-many relationship, and this complexity comes to the interpretation and reporting, figuring out which biomarker fits which need for the patient. So if we take these three factors together, which is the heterogenic classes of genomic alteration, as well as the many-to-many relationship of these hundreds of biomarkers, and the different methods, each one with its own sensitivity and specificity and limitation and cost and turnaround time, all this together make it a complex approach when we are discussing myeloid neoplasia testing algorithms. And this is the end results of such complexity. And I'm sure all of you have seen multiple number of testing per patient. Some comes from FISH, some come from cytogenetics, flow cytometry, molecular studies, and we end up having multiple tests at multiple time points to help the patients, which make it a little bit complex. And in most places, usually these done at different laboratories where they don't necessarily directly do it as an integrated, so everyone is doing their own job without knowing what the other lab is performing, which make it difficult to actually verify the results, discuss what cytogenetics means to the molecular result or the flow cytometry. And usually all this dumped into the electronic health record, where it's become the burden on the hemato-oncologist to combine and integrate all of these data into the risk assessment and choosing the right therapy for the patient. So this paper, no wonder why this paper creates a lot of optimism in solving this complexity and simplifying the workflow. And what they are proposing is simple on paper. They are basically saying, you know what, let's sequence all the whole genome bases, which about 3.2 billion bases. And by sequencing almost 98 of them, it's considered a full sequencing. What about the 2%? The last 2% is usually a repetitive region, the centromeres, which is usually void of genes and difficult to sequence. And hence that is not a 100% sequencing, but it's close enough. So in this paper, they use this method, which is the ultimate sequencing method that we have today when it comes to screening DNA. And they try to see how's this method compared to cytogenetics and FISH in detecting all these abnormalities and biomarkers. So I will use this figure to explain the landscape of the complexity. We will have the morphology assessments all the way. It's not going to disappear. This paper has nothing to do with the morphology. It's assumed it will always be there, as well as the flow cytometric immunophenotyping. But the story is different when it comes to the karyotyping and the FISH panels. So as we stand today, we are at this stage. We are here today at point A, where we need all these 5 modalities to actually give the answer to the patient. And the molecular single genes is usually more than 10 different tests, the FLIT, the NPM1, JAK2, etc., that we have in the lab. Each one is done on a different methods, require a specific amount of the DNA or the RNA, and it takes its own pathway. And the paper at the molecular whole genome sequencing, which is the lower row, where it's basically say we want to move from point A to point B, which is, let's call it the promised land, where life is much simpler. And basically all we need is, beside the morphology and the flow cytometry, just do the molecular assay, the whole genome sequencing, and we will replace karyotyping, FISH, and molecular single gene assays. So the paper was performed in Washington University on 263 samples, and they divided in 2 groups. The first group was retrospective, 146, and 117 prospective new cases. In the retrospective cohorts, they had 107 AML and 39 MDS. And out of those 107 AML, 87 of those had successful cytogenetics analysis. They used it to compare with whole genome sequencing, and 20 had unsuccessful cytogenetic analysis. While the MDS group had, all of them had successful cytogenetic analysis. For the prospective samples, 68 were AML and 42 MDS, and the cytogenetics was much more successful in the AML group. Only 4 of them were unsuccessful, and similarly in the MDS. So the first thing they did is they found that through karyotyping and FISH, 40 recurrent translocation and 91 copy number variants, all of them were detected using whole genome sequencing. So the correlation is 100% for those biomarkers. What is more interesting is they added new findings in 40 patients, which represents 17%, using the whole genome sequencing only. And in the prospective study, they finished on average the whole genome sequencing in 5 days, which is extremely impressive, knowing the difficulties and the challenge around the whole genome sequencing. It's a very impressive and remarkable achievement. And the new finding occurred in 25% of the patients. And this changed the risk category for 16% or 19 patients. And this is absolutely amazing when it comes to a single test that add all of this value to those patients and only within 5 days. So they listed the retrospective cohorts as well as the prospective cohort and divided them based on the genetic risk group based on Ellen. And you see here is the favorable risk, whereas some of the patients shows RUNX 121 gene fusion and biallelic CBB alpha and other biomarkers. I will not go into details of each of these group. Here is the intermediate risk in the retrospective and prospective, as well as the adversary group, which shows other biomarkers with complex karyotyping. And the idea here is this method performed so well in the retrospective as well as the prospective and selected patients. So one of the things they focused on was the structural variation. And you see here is the gray bar represents almost 183 structure variation or translocation that's picked up by the cytogenetics or the FISH. And they through whole genome sequencing, picked up another 13 patients, and they confirmed all of them through FISH, PCR, or RNA sequencing. And that showed that it is an actual true finding. So the question now becomes, how do we replace cytogenetics, which is under the microscope, where we see the translocation between chromosomes and the NGS, the sequencing? So apologies if this sounds basic, but just to make sure that everyone on the same page, I will have only a couple of slides to show the technical, how do we move, how do we detect structure variation using sequencing? So when we have the sample, we have to fragment it into millions of short DNA segments, usually less than 300 base pairs. And there are multiple technologies in the market. One of them is Illumina, and I'm not showing it here for any reason other than it's one of the most commonly used methods. But Thermo Fisher, MGI from China, as well as BacBio are very well considered alternatives. So in Illumina specifically, they have a flow glass where they bind the DNA vertically, and consider or imagine there is billions of these DNA fragments, short fragments in the glass. And using a laser, they will start doing a synthesis and complementary for these segments. One by one, they start generating base calling, which eventually will appear as a text similar to these 5 reads. But in reality, these are billions of reads. So when we look at this, initially, it does not mean anything to us. It's a random sequence. It is short. I have no idea what it means for the patient or the biomarker. So the bioinformatics will take all this data, which is in billions. And actually, in this study, the average data that's for each patient is more than 120 gigabytes of data. So the bioinformatics will start with read mapping. It will take every read and scan it through the whole genome reference, trying to figure out where does it map. And once it's find the location, it will assign a value. It will say this reads belong to this site, and it will does all the reads sequentially. So the next step is we are trying to pick very unique reads. That is, half of it is matching the genome and the other half does not match. And in this case, you see the blue color, which matches the reference genome, while the red color sequence does not look like the reference genome. So at this stage, when we pick up those split reads, we ask the question, where does the red part come from? And we try to remap it. And we find that it's actually, for example, RUNX 1T1 gene. So these 5 sequences will tell us that they belong to different location at the genome. And hence, this is how we detect structural variation using the NGS. On the karyotyping site, it's done visually by the microscope, looking at the translocation, but we never know exactly where is it or the actual base pair. Usually it's within 1 million or up to 5 million resolution. The second part beside the translocation was the copy number variation, which is deletion or duplication of large segments of the DNA. And in this study, they picked 83 of those. 14 of them were actually completely new and missed by the standard clinical practice. And they confirmed those 21 new copy number variation using arrays, FISH, and sequencing. And they mentioned in the supplementary one of those deletion, which you see here, and it's a very tiny deletion with a green color that represent 13% abundance. And this would be missed completely by karyotyping if it was done, because it's so small the eye cannot see it. But the whole genome sequencing, given that it's read every bases, we're able to detect this change in the coverage at that region, and hence it was able to detect a new mutation at this location. So let's talk about this impressive 5.1 days from receiving the sample to issuing a report. given that this is one of the most difficult assays that we have, and extremely complex with a huge amount of data to analyze. So their practice is to take the tumor sample, do the DNA preparation and extraction, and they start making what we call library, NGS library in the lab. This process from extraction to making the library takes 8 hours in this study. And once it's done, this is a short segment of DNA ready for sequencing. It goes into one of the Illumina machines, the big ones, and it takes about 26 hours to sequence it and to produce about 120 gigabytes of data, which represents that every position in the genome is covered on average 60 times, which is the 60x that you see in the top of this figure. After that, they take the data into automated analysis, which I think is the key part to the success of this project. Nothing is manual, everything should be automated if they want to handle this large amount of data. And they issue the reports, and the whole thing takes about 3 hours, which most likely is automated reports. So they listed their patients from April to February, and you can start, you see from the left side that's the left half of this figure is some of the samples reach 10 days, and in some cases more than 3 weeks. The green color represents the library preparation and the quality control, which is not usually the big problem. Sequencing was fixed. The blue color is always, except for one case, was fixed, and this is 1 day. But the analysis and the reporting was variable. Initially, it seems like it took them a few days per case, but as they develop more experience, you see from September onwards that the time for reporting is extremely small, indicating that they have learned enough from the first half of the cohorts, and it's become a streamlined clinical service. So now the question is, can we rely on the whole genome sequencing for the risk assessments in compared to cytogenetics and FISH? And for that, they are showing the risk assessment based on the conventional risk stratification for 71 survival patients with AML who were treated with chemotherapy alone after remission. And you can see it's divided them in favorable, intermediate, and adverse group. When we compare it with the whole genome sequencing, we found that the agreement is almost 90%. Only 8 patients needed to change their risk assessment. And those 8 patients, as you can see in one of the supplementary figures, the top row is the whole genome sequencing, changed 7 of them to adverse from being intermediate. And only 1 of them was favorable after it used to be intermediate risk. And below, they are showing in the red blocks what is the new risk defining information that came from. So for example, in the third patient, the third column, you can see there is a translocation that changed the risk in 3 of those patients. Interestingly, 27 of the patients has unsuccessful cytogenetics, and because undetermined, this was the risk, the overall survival. But the whole genome sequencing was able to actually differentiate them into 2 groups. The purple color, the not adverse, which is the intermediate and favorable, compared to the adverse group. Which means that even in patients with unsuccessful cytogenetics findings, whole genome sequencing was able actually to provide risk stratification information. So now, after this impressive findings from the study, let's talk a little bit about the challenges with whole genome sequencing as of today. And I listed 3 major challenges. The first is the big data, the second is the cost, and the third is the logistic. So what do I mean by big data? So you see here is one of NASA published space images from the Hubble telescope, which is known to be huge in terabytes of size, they transmit it from the space to Earth for studying. And the data acquisition in 2025 expect that the astronomy images will mount to 1.5 or 2 exabytes of data. And I will tell you what exabyte. It's different than terabytes, much larger, and we will see it in the next slide. Interestingly, in just 3 or 4 years from now, YouTube will have more data than the astronomy or all the telescopes in Mars or Hubble or others. So it will almost have 1 exabyte more than the astronomy. If we look at the genomic data, we're expecting actually way more than YouTube data, more than 21 exabytes. And this tells you that genomics is going to be the major data generating field around the world. And exabytes, for those who's interested, is 1 and 18 zeros behind it, which is unthinkable of today's capacity. And this means if you want to go the genomic full force, we are expecting to be big data heavy with a lot of infrastructure to handle all of this data as it becomes part of the clinical surface. And I'm not talking about the hemato-oncology, you are talking about genetic disorders as well as gynecology, reproductive health. Whole genome sequencing is a disruptive technology that is changing almost all the branches of medicine. And with that, the paper published in 2015 by Stevens and his group, they were actually suggesting that we should change big numbers when we are trying to exaggerate and we say arqam falakiya or astronomical, is actually we should start using genomical, and I agree 100%. The amount of data produced from those machines is mind-boggling. Which brings me to the costs. So it's been known, maybe you have heard, a few years ago that there is a huge promise that we will reach very soon $100 for a whole genome sequencing. And the white, the green color is representing the cost of sequencing for the genome. It used to be in hundreds of millions in 2001. And today it's reaching around 1,000. So we are not yet there at the 100 promise. And you can see there was a drop in the sequencing until 2015, and then there is a plateau. And in my opinion, it's not dropping because the number of companies who's able to provide the service is very limited. And probably there is, some monopoly effects that take place in here, which prevent us from going to the $100. So the paper mentioned that the cost was around 1,900, but that is still expensive. And it is much cheaper in a huge academic center like Washington University, where they have special discount. They are in the US, very close to the vendors, which is Illumina in this case. So you can imagine in outside the US, for example, in Saudi Arabia, we would expect 30 to 50% increase in the price because of the shipping and the local distributor fees. So we are looking at $3,000, and we have not mentioned the bioinformatics or the storage analysis, nor the interpretation and the overhead. So realistically speaking, and this is just my opinion, I think if we have done this project in the country, it would cost almost $4,000, which is still significant, which almost 17 to 18,000 riyal per patient. So cost is still a major issue, despite the fact in the paper it mentioned it's relatively cheap. And beside the cost, I think the logistics, especially in this country and probably in the Middle East, not just Saudi Arabia, is causing huge challenges to adopt the new trends of testing. So in our experience, purchasing through NIPCO for such projects would take us a year to a year and a half. Importing the reagents from the US will take half a week to 2 months, sometimes depending on the holidays and the content of the kit. Sometimes there is risky, dangerous materials that need to be have a special importing permits. And finally, the local support from those companies, Illumina, Thermo Fisher, MGI. Usually, if the machine is filled, it will take, depending on replacement, 2 to 4 months. So together, these are in no way or mean is criticism for this great organization, rather than telling you the actual reality in the country. It takes time to adopt such things, but by no means it's not a block, and we are seeing improvement in all aspects as we move forward. So I hope I have not put you down with these limitations, whether it's cost, logistics, or the big data. And the question now is, knowing what we know today, should we give up on the whole genome sequencing? Before I answer this question, let me remind you that the alternative of whole genome sequencing is focusing on the coding region, or the whole exome sequencing. And this is almost 50 million bases, which represent 1.5% of the whole genome. We can go even smaller to a small NGS panel, where we focus on actionable genes for the myeloid, and they usually represent 1 million to 500,000, which is a small percentage of the genome. And this could represent a fantastic alternative meanwhile. So where do we put these panels? We saw point A, and now we also saw point B. By adding molecular targeted NGS panels, we could actually walk slowly in the middle part and simplifying the workflow. Today we have DNA panels, RNA panels, and there is also integrated panels which has both. DNA is good for mutations, RNA is good for structural variation. And by combining both, we actually could end up, by combining both, we could focus on the CMP, the single mutation, the insertion deletion, as well as the translocation and some of the copy number variants. And at National Guard hospitals, we have implemented DNA panel with 84 genes and RNA panel with almost 75 genes. Which is good for gene expression, gene fusions, as well as point mutation, insertion, deletion, including the FLT ITD mutation and CBP alpha, which is notorious for sequencing because of the high GC content. So does target panels have any leverage or extra pros when it's compared to the whole genome sequencing? And the answer is yes. I took one of the supplementary figures in the paper where they compared the targeted sequencing to the whole genome in terms of sensitivity of the mutations. And you can see the first column is the NPM1. The blue color represents it was detected by whole genome sequencing. The red color are one case was missed. So sensitivity is 97. It's great for NPM1. But let's have a look at the CBP alpha. It's 83. They also missed one patient. When it's come to FLT3, they missed 3 patients, so it's not that great. The ASXL1 is great. They missed 1. RUNX1, they missed 3, so the sensitivity is 82%. While with the TB53, which was surprising, they missed 4 patients. And when they compare it with the PCR, which is one of the gold standard method called FLT3 ITD, they missed 4 patients. So it seems like smaller panel with higher coverage and focused targets is still more sensitive than the whole genome sequencing, but it will miss, it's not as comprehensive, but there is still a value for it. So if the question now become, this is one example why whole genome sequencing missed some of the FLT3 ITD. In the top, you see the normal FLT3 structure between exon 14 and the exon 15. When there is tandem repeats after the exon 14, this sequence is duplicated, and usually the size could range from 15 bases to 300 bases. And what happens if it is too long, longer than the whole genome reads, then we start missing some of these mutations. So this is a limitation because the read length is shorter than the actual ITD mutation. If the ITD is short, it will be easily picked. But once it's in excess of 50 or more, then we start having some difficulties in figuring out how big is this ITD. The smaller panels, it has a fantastic smart ways of dealing with the ITD, and it's usually much more sensitive than just using whole genome sequencing. So this is one of the limitation of whole genome sequencing. So this is where we think we will situate the molecular targeting NGS panel. It is great for combined with morphological and flow cytometry. It will reduce the need for FISH. Definitely, it's so good with fusions. Karyotyping will be still needed, and this is why it is still less simple, more complex than the whole genome, but it's not by far. It's just away from the whole genome sequencing, in my opinion, by 2 or 3 years. And with that, I reach to the end of my talk. I would like to thank the Department of Pathology at King Abdulaziz Medical City, Dr. Al-Ajlan, Dr. Arij, and the HemBath team, as well as the adult and pediatric hem-oncology team, Dr. Al-Asker, Dr. Ayman Hijazi, our staff at the Molecular Genetic and Cytogenetics Lab, Prof. Mohammed Abloui, as well as the staff in Translational Pathology Lab. Thank you very much for listening, and I'm happy to take any questions.
Okay. Thank you, Dr. Saeed, for your elegant and comprehensive talk today. And I hope that at one point of time, we can adopt the whole genome sequencing for our hemato-oncology workup, especially for some of the cases where we have unsuccessful cytogenetic culture due to technical failure or culture failure. For example, one of the diseases is really multiple myeloma. We are looking for more comprehensive genomic test to help in risk stratification of these patients. I guess just I would remind our audience to please type your question in the Q&A section. We received one of the questions from our audiences. The first question is, how accurate is whole genome sequencing for FLT3 ITD with long repeats? And great presentation, thank you.
Absolutely, a great question. I think you wrote it before we spoke about the FLT3. And just to iterate, it's less sensitive than the targeted panels. 76% sensitivity and 90% when it's compared to the PCR methods.
Okay, great. The second question, does 60x coverage suffices for report small clonal size, i.e. less than 5% allele frequency?
Fantastic question, and I think this is a personal, opinion. 60x is not great at all for detecting ultra rare mutations. We need, on average, we sequence more than 500 to 1,000x in order to pick up those low allele frequency. So, to answer your question, 5% of the 60x is only 3 reads. And to me, 3 reads to call a mutation is not that great. So probably the limit of detection will be 10 or 15 or 20%, where we have enough evidence to support this mutation. So I don't think it's that great for the ultra rare mutation. But remember, the whole genome sequencing here will be used for newly diagnosed patients, not treated yet. So we're probably going to see high allele frequency for those mutations. But definitely it will not, the whole genome sequencing will not be good for MRD. Otherwise it will cost tens of thousands of dollars compared to the targeted panels.
And speaking of MRD, I think we have a question here. Can we use whole genome sequencing for detecting minimal residual disease?
Only if you have $1 million and above for one patient. If not, then it's not cost effective at all. It's always better to go to smaller panels. And even when we know the mutation, let's say we sequence the patient with whole genome sequencing, we picked FLT3, now we know this patient has it, so now we will do MRD by PCR. This is a cost-effective approach instead of doing NGS.
Great. We have one more question here. Again, about MRD. For MRD detection, which technique would be better, PCR versus NGS?
Right. So I did not speak about the small details, the technical details for NGS, but NGS has a lot of different types. We have, beside the whole genome, we have hybridization methods, we have amplicon methods, and basically these are fancy names of how we capture or amplify the targets. And some of the companies, they add something we call unique molecular barcodes. These are random sequences that we attach to every read that will allow us to trace the MRD in a very sensitive way, eliminating the noise. But it comes with the cost that we need to increase the sequencing coverage. So patient who will cost, let's say, $1,000 will cost $3,000 for MRD, simply because we need a higher depth to detect those. So having said that, which one is more sensitive? It would be difficult. BCR also is different types. There is digital BCR, the new methods. There is the standard real-time BCR. Digital BCR can go to less than 0.001. The standard BCR probably 1%. So, you know, there isn't a harmonization or a guidelines clear for the MRD. I think it's being developed as we move forward with more patients and more convergence of the technology. Because one of the problem, it's a blessing and a problem in the same time, having a lot of molecular methods. And this makes it sometimes difficult to compare apples to oranges. In our practice, what we plan to do is NGS panels for the newly diagnosed patients, and then the follow-up for MRD will be done using real-time BCR to 1%. As we move forward and we want to go to even a more resolution, we will adopt the digital BCR for MRD.
Okay. Okay. We have one more question here. In both stem cell transplantation to check chimerism of donor myeloid and lymphoid cells, could we use whole genome sequencing for checking chimerism percentage?
I mean, you could, but it's 3 billion bases. Chimerism analysis will rely on 16 loci or almost, I don't know, 200 bases. So if it is cost effective, it's always go to the chimerism, which costs, you know, less than a 500 riyal compared to 18,000 riyal. So I would just go for keep the chimerism using the fragment analysis or the microsatellite analysis.
I have a question, Dr. Arij, can I?
Yeah,
well, great and excellent talk, Saeed. This is as usual. Now, and you presented already the obstacle, the logistics and the obstacles and the challenges, you know, with this technology. Definitely, I mean, doing whole genome would be nice, and you have the panel, and depends again on how extensive the panel would be and the cost and etc. Is there so where is the cost lies? Is it in the like in the machine, in the kits and the analysis, etc.? Is there a way to approach this and dissect this and see how are there alternatives, competitors with the other companies? You think, you know, entering the market, would that lower the cost? I mean, how do we see the future of this, and how long will it take?
Great question, and thank you for your kind word, Dr. Ahmed. So I think the most of the cost is actually the sequencing on the machine itself. It's not the library, it's not the QC or the DNA extraction, it's the sequencing. Beside the sequencing, I think the storage of the data itself will start over time, will be larger than the sequencing. We are going to reach a stage where it's going to be cheaper to delete the data from the server and just repeat the sequencing if you wanted it, instead of keeping it for long term. This is how actually the sequencing cost is reducing. Having said that, the major players when it comes to whole genome sequencing is Illumina and the MGI, which is a branch from Beijing Genome Institute. This is a Chinese company. They bought a technology from the US. It's used to be called Genomic Complete or GC. They bought it, they modified, they built their own machine, and they are now competing with Illumina. And I was very excited a year ago when they came into the market, thinking that the price will be at least 50% less than Illumina. The last quotation I got was 3 days ago for this presentation. I took, quotation from both companies, and unfortunately, they are comparable. They both are expensive, and the prices are similar. So that was a little bit disappointing. So how low we can go? In honesty, just to not to grill Illumina, the price they gave us here today was not far away from what they give in the US. Basically, in the paper, they mentioned the gigabyte is $10, and the price I got was $7.5, which is cheaper than the average in the US. But Washington University, I think they can reach to $5 because they order a lot. So it's become, I think we are reaching the lowest, price as we speak today. How to improve this? I would imagine if we have a large scaling program for the whole country, where we sequence hundreds or thousands of patients actually, then we might have a very good deal from either companies where we can reduce the cost by probably 30 up to 50%. But that's just my opinion. And even if we solve for the sequencing, the other cost comes beside the storage is the interpretation. So you need, if we don't adopt the automated reporting system, which I think is a key point. I have no idea how easy it was for Washington University to develop the pipeline, but it will take a while to develop and capture all these biomarkers with all this very difficult region to figure out the FLT3, the CPP alpha, the translocation, the RNA, the DNA. It's just a lot of mess. It requires a lot of work. But over time, I don't think the analysis will be a huge problem. Pipeline will be published. It will be open source. It will be used. So we will always circle around between sequencing and storage cost. In my opinion, I think we can reach to a very reasonable price within 2 years from now, where we can, the cost of whole genome will be much cheaper than doing the cytogenetics and fish panels. And we already seen this using NGS in solid tumors, for example. The panels today is cheaper than doing the sequential single gene testing by 40%. But it took, lung cancer started early enough. The sequence dropped faster. And I think you will see same pattern here with the whole genome sequencing.
Okay, I have more questions here. The second question is, when the whole genomic map will establish for GCC Arab nationality, and is that and this would be would it be cost effective?
That's a good question. I think there's multiple genome projects ongoing at different institutes, including KMARC, التخصصي, KAXT, as well as KAUST. But these usually are research projects, not yet clinical. I think in the future, this will be a common surface. As we speak today, it's not yet a clinical practice for the whole genome sequencing, but we are seeing the tantalizing evidence. And I hope the community and the society and all the stakeholders will think outside the box and see how can we actually adopt such methods for the larger good of patients.
Okay, one more question. Whole genome sequencing will generate a huge data, as you said, and there will be a lot of significant variants, i.e. pathogenic versus BOS with high coverage. What you will report, and giving the reports with multiple variants, some of them could be not listed in WHO or any guidelines. Wouldn't that be confusing to the clinician and the pathologist?
A very good question. I agree. On average, we will see 4 million mutations per patient. A lot of them will be BOSs never seen before. But since this is a clinical test, we will always go for hotspot mutation. So only hotspots, non-actionable mutation would be reported that is well studied. And then the question becomes, what do we do with the rest of the BOSs? And to answer this, we simply keep an eye on those mutations. It's a research actually findings, and we will accumulate more knowledge about them as we move forward. So the more we sequence, the more we understand about those BOSs. And with automated pipelines, we can go back automatically for any BOS and figure out every 3 months if there is any new evidence that can push it to the clinical settings. But usually clinical reports will not include BOSs, just to reduce the number of pages. The pages of our standard NGS report is 5 to 10 pages on average. I can imagine with whole genome sequencing, we can go for 20, 30 pages, and no one has time to read this, or even it's not practical. So only hotspots, I would say.
Okay, I guess this is one comment. Fantastic talk, Dr. Saeed, and they said, do you perform, and they agreed that the use of targeted panels instead of whole genome sequencing for genomic diagnostics. Do you perform paired tumor and germline testing or germline variant detection and identify acquired somatic mutation associated with the disease progression or relapse for MDS AML patients?
Great questions. As of today, we don't do germline simply for the cost effective. This is the only reason. The second reason is if we focus on the cancer cells, using clever methods with the bioinformatics, we can pick up those somatic mutations. So any germline will be disqualified, and this enables us to focus on the hotspots. If you have the capacity or the money, then by all means, germline and tumor is always good, but it's not practical given the high cost of these assays. And thank you very much for your kind word.
Okay, one more. I think this is would be the last question maybe. With transformation and vision of 2030, does they adopt molecular testing? From Dr. Shahrani.
Absolutely.
So shall I repeat the question?
No, I read it. Thank you, Doctor. I see it. So I believe a huge vertical of 2030 vision is the healthcare for the citizens. And I think, genomic profiling is the heart of precision medicine. Basically, we are tailoring the risk assessment, the therapy, the diagnosis for every patient based on their genomic data. And this represents one of the best examples to be adopted. And I think more hospitals are getting into the NGS area. التخصصي الحرس already started. King Fahd Specialist Hospital in Dammam, as well as King Abdullah Medical City from MOH in Mecca. So I think there is a transition toward these tests.
Okay.
Before we conclude, can I also the rest of the time, Saeed? So if you have let's say the capabilities and you wanted to move ahead with utilizing such big data that will be generated, and integrate it into machine learning and artificial intelligence and etc. So what would be your, let's say, dream?
I would say, for example, we'll start with a couple of NovaSeq machines, and this is probably 10 million riyals. And then the library for each patient will cost, roughly speaking, let's say 2,000 to 3,000 US dollars. And if we aim for 1,000 patients a year, this is 3 million US dollars. So you are talking about an initial capex of 10 million riyal and running cost or OPEX of probably 3 million US dollars or I don't know, 12 million riyal. And for a national program, this is not much really, given all the benefit that comes out of this. And I think, at least in my experience, we are not that far behind. It's really the simple logistic that it can be fixed, and the collective efforts from the society and the stakeholders. This can be done, I would say, within a year.
And you would likely integrate the genomic data with the clinical data and trying to come up with, whatever algorithmic, automated interpretations, etc.
Absolutely.
I'm thinking big, and I think that's where we're moving ahead in terms of this technology. Having these high-performing computing and others, bioinformaticians, etc., capabilities.
I absolutely agree, and I think having the automated pipeline would be before we actually start testing the patient or getting those machines, because this is I would say, the core key for the success of this project. Because sequencing is now become routine. It's not, I think, the trickiest part would be the pipeline itself and making sure that it's correlate with the FISH, with cytogenetics and other tests. But it's very doable, given the fact that today we have pipelines for the NGS panels, which is not far away from what's used for the whole genome sequencing. It's just smaller. We just need to scale it a little bit up, but it is very doable.
Great. Thank you very much for wonderful talk and wonderful comment and questions. I guess we reached to the end of our time. I would like to thank Dr. Saeed, our speaker again, the respected audiences, the sponsoring company, and just to remind you again, please stay tuned for the next SSBMT webinar, which is going to be about how I treat PNH by Dr. Austin Kozalaraj, I guess. Hopefully I say the name correctly. And thank you and have a great night.
Great, thank you.
Thank you. Take care.
Thank you, our audience.